Hello, can somebody please confirm that you're hearing me?
I can hear you.
Great. I'm a minute late here because Zoom updated itself when I tried to open it. So it downloaded something and updated it, and that took a few minutes. Are there any questions before we begin? All right, I'm not hearing any questions, so let's go ahead and begin. Should be able to see green now. All right, so today is July 19th, and we should be discussing chapter 3 and ending observing microorganisms in chapter 3, and then beginning chapter 4. Any questions about any of that? Do note that you do have your first quiz this Wednesday, quiz 1, and it'll cover chapter 1 and chapter 10 and the start of chapter 2. So it will cover what we covered on Tuesday, but not what we covered on Thursday. Any questions about any of that? The quiz will also cover labs 0, 1, and 2. So lab 00, lab 01, and lab 02. Any question about the quiz? Remember, there will be 2 quizzes you have to take. Quiz A, which will be essay questions and short answer questions. And this one, once you start it, you can have until 11:59 p.m. on Wednesday night to submit that quiz. So it's not exactly timed, although it does need to be turned in by 11:59 p.m. on Wednesday. And you can take it, I don't know, midnight Wednesday morning, or maybe it's 12:01 Wednesday morning. So you have almost 24 hours, maybe minus a minute, to take that quiz. And like I said, it's not timed, although it must be submitted by that time. Quiz 1B will be multiple choice questions. And once you start it, sometime between midnight and 11:59 p.m. Wednesday, it is a timed exam and you must complete it within the time frame. I know you have 1 minute for each question, and then you have like 3 or 4 extra minutes to work on the quiz. So it is a timed exam. It's timed so that if you start looking up the answers, you will be able to look up 1 or 2 questions in your notes or in the textbook. However, if you spend more time on either 1 or 2 questions looking up the answer, you will then run out of time on the quiz. And any questions you do not answer will be marked wrong. So it is time to prevent you from really looking up the answers. So you do need to know the material before you take the quiz, because you will not be able to look up more than 1 or maybe 2 questions. Any question about any of that? All right, if not, let's move on to chapter 3. And before I get started, let me state that I did do the grading So I got the lecture worksheet for chapter 2 grading. And I actually graded that a little bit before it was due. And so you had about a day to make corrections. But normally in the lecture and the lecture worksheets, you will not be allowed to redo those answers. And so if you don't know the question, you should look up the answer because you do want maximum points on the worksheets. Any question about any of that? All right, and then I also graded, what did I grade? Lab 2 and 3, I think. So they are graded the first time. And if you want full credit and you didn't get full credit, then you can resubmit answers by this Saturday. And then I'll go back and regrade those. I have not regraded Lab 00 and Lab 01. I was actually hoping to get that done this weekend and I ran out of time. That frequently happens where I will put priority to the first grading. And because we go so rapidly in the labs, I am very late sometimes in doing the second grading. But hopefully this week I'll get the second grading of Lab 00 and Lab 01 graded. Any question about any of that? Now remember in the labs, if you get 0.01, that is not your grade. That's a code for me to remember, or a code for me to tell me to go back and see if I need to regrade the answers. So the first time I grade it, everyone will get either 0.01 points or they'll get full credit for the assignment. And then the second grading, you'll get the grading between 0 and full credit, which is usually 5 points. All right, if there's no questions, let's move on. We talked about bright field illumination and discussed how it was different from dark field illumination last time. Any questions about any of that? Remember, on dark field illumination, you only need to know the concept. You don't need to know every detail of it. However, bright field illumination is what we normally use with the compound light microscopes in the lab. It is bright field illumination. So you should know the details. Now, electron microscopy is a little different than light microscopy in that we do not use light to view the object. We use electrons, or you could say electron waves, meaning electrons are both a particle and a wave, which actually is true for light, but we won't get into that. And because the electrons have a much smaller wavelength, we can magnify an object many more times than we can magnify the object using light. Any question about any of that? So electron microscopes use a beam of electrons instead of light. And then we use electromagnetic lenses, they're not glass lenses, to focus the electrons. The shorter wavelength of the light, which is 1,000 times smaller than visible light, gives greater resolution, and then we can magnify at least 100,000 times more than we can with the light microscope. We can magnify things like viruses, internal cell structures, and then molecules that you cannot see with a light microscope. And here are two images of objects seen from the electron microscope. The first is with transmission electron microscope. And as you can see, we're looking at a plane or single slice of this cell. And we're only looking at that. Let me blow that up. We're only looking at this in 2 dimensions, meaning we're seeing a single slice through the cell. And so if we wanted to see the top of the cell, we'd have to do the slice above this near the top of the cell. And if we wanted to see the bottom of the cell, we'd need to do the slice through the bottom of the cell. And that is transmission electron microscope. That differs from the second main type of the electron microscope called scanning electron microscope, where you see the surface of an object. And as you can see from this picture, it's in 3 dimensions, whereas this one's in 2 dimensions. Any questions about any of that? These are the 2 main types of electron microscopes. Scanning electron microscope, giving you the details of the cell, and then the scanning electron microscope showing you the surface, in this case of the cell, or the surface of an object. And you can see the object in 3 dimensions. There are other types of electron microscopes, but those are the 2 main ones. So scanning electron microscope, where we see the object in 3D, and that's some crystal. And then transmission electron microscope. I think this is of the chloroplast, where we're actually seeing the membrane of the chlorophyll. Another form of electron microscopy that you do not need to know, because we're not going to be examining it, is tunneling microscopy, or scanning tunneling microscope. It uses a metal probe to scan a specimen and allows the resolution of 1/100th of an atom. So it really allows great magnification. And here we're seeing the protein from E. coli. A 4th type of microscope, electron microscope, is atomic force microscopy. You're also not going to be examined on this. It uses a metal and diamond probe inserted into the specimen and then produces a 3-dimensional object when it's viewed. Here we're looking at the toxin from Clostridium perfringens. All right, any questions about the electron microscope? If not, we're going to move on. We're going to talk about preparing specimens for a light microscope. And when you're looking at these cells here, which of the cells are easier to view? The ones on the left or the ones on the right? Come on, somebody's got to give a,
The ones on the right.
it's pretty simple.
The ones on the right.
The ones on the right. The only difference between these cells on the left and the cells on the right are the cells on the right are stained with a dye. And that causes the cells, when they take up the dye, to have higher contrast with the background of the slide, making the cells show up better and giving greater resolution. So cells have little contrast with the background of the slide, and that makes them difficult to observe. And so if we use a stain, they're much easier to observe. And that is the primary reason for using a stain, simply to make the cells show up better. And the only way the stain is doing that is increasing the contrast of the cells with the background of the slide. Any question about any of that? All right. So with microscopes, most microbes are colorless, and that's why they have little contrast with the slide or the medium. And when you view that, you know, there's not much difference between this cell here and then the background, which is a glass slide. And there might be media or water in here too, and there's little background contrast between the media and the cell. And so to see them, we use staining, or there is another thing you could use, a phase contrast microscope, and we haven't talked about that, but it allows you to change the light so that the cell can show up more distinctly from the background. And we did discuss dark field illumination right here. And this allows the cell to show up much clearer from the background. And phase contrast microscopy is not the same as dark field illumination, but it's similar, meaning you look at the cells and it's similar. Before you stain a cell, you do need to make a bacterial smear. That's a thin film of solution of microbes on a slide. And what you do is you get your glass slide, put on a drop of water. I don't know if you can see this piece of paper or not. And then put on your cells into the water and then spread the drop of water all over the slide. And how you make a thin smear is you use very few cells, just amount of cells so you can barely see the cells. And you don't use a loopful of cells, you use, like 1/20th of a loopful of cells, meaning you can barely just see the cells on the loop. And then you allow the smear to air dry, and they're usually fixed by air drying and then heat fixing them, passing the slide over some heat, which when we do it, it's usually you put them over the Bacto incinerator, and that tends to kill cells. It does not kill all cells, but many bacteria will be killed. Endospores would not be killed, but many other bacteria are killed. And like I said, we usually apply air drying and heat fixing. And then we take that bacterial smear and then we stain it. Do understand that staining is not making the bacterial smear. And when you do a bacterial smear, you're not adding stain. Students frequently get that wrong when I ask a question about that. Stains are simply dyes, and they're salts consisting of a positive and a negative ion. And where the dye, the colored ion is, we call that a chromophore. That determines what type of a stain it is. If the chromophore is in the cation, the positively charged member of the salt, then it is a basic dye. If the chromophore is in the anion, the negatively charged portion of the salt, then it is an acidic dye. Any questions about any of that? Bacteria have a slightly negative charge, and therefore basic dyes do stick to the bacteria. And generally that gives slightly better staining. And this is most of the stains that we use in microbiology: crystal violet, methylene blue, malachite green, and safranin. And we'll talk about each of these later in the lesson. Some of them we'll talk about today, like I think we talk about crystal violet and safranin today. Malachite green we might talk about today as well. Acidic dyes have a negative charge, and so they do not stick to the bacteria. They typically stain the background and not the microbes. And some dyes, which are acidic dyes, are eosin, acid fuchsin, and nigrosin. And they're sometimes called a negative dye or a negative staining, where you stain the background of the cell instead of staining the cell. And this negative staining term comes from photography. If you knew the old films, what do you call that? I don't remember. You would be given a negative, and then you would develop the pictures from the negative. The negative would be stained opposite from what you're seeing. So in a black and white photograph, the person would be black, and then maybe the light would be, think about that. No, the light wouldn't be light. The light would be black, absolutely black. And then something that was dark would stain light. And that's why it was called negative. And that's sort of what the acidic dyes do. They stain the background, making the background dark. And then the cells are not stained, and so they're light. Why it's called negative staining. Any questions about either acidic dyes with negative staining or a basic stain, basic dye? So here you have a basic stain staining the cells, and they're very visible. And then here we have a negative stain or an acidic stain, and it's staining the background. And when you stain the background, you can see the cell quite readily. Any question about any of that? All right, if there's no question about that, let me state that this is a simple stain where you're using one dye to stain the cells. And there's no difference that we can see, at least in the coloration of the cells when we're doing a simple stain. Meaning all the cells here stain reddish, including the cocci and the bacilli. And then the same thing here with a negative stain. All the cells stain the same. In this case, they're all light and the background is dark. Let me mention that when we stain microbes, there's three types of cells. There's a simple stain, which generally uses only one dye. There's a differential stain, which shows differences between different cells, especially different types of cells. And usually a differential stain uses two or more dyes. And then the third is a special stain where we're applying the dye to stain something specifically in the cells or to illuminate a specific characteristic of the cells. Like when we're staining for flagella, it does not stain or does not show up with the light microscope with the simple or differential stain. We have to use a special stain to show up the flagella. And there are many different special stains to show a specific characteristic of cells. Like you could do a stain to show starch in plant cells. That would be a special stain. You can use a stain to show endospores in cells. That would be a special stain. Special stains can be staining many different properties of the cell, and they'd be a different stain, but they can be many different things. All right, any question between the three main types of stains? A simple stain, as I stated, is an aqueous or alcohol solution of a single dye. Its primary purpose is simply to increase the contrast between the background of the slide and the specimen we're observing. In this case, the microorganism. And you can have a simple stain being either an acidic dye or a basic dye. Okay, I think your textbook actually got that wrong. It called a simple stain a basic stain, and that's not true because a simple stain can also be an acidic stain. The point is you're only using a single dye, and so all of the cells are staining the same. When you stain the cells, you apply the stain to the cells of a fixed smear, meaning you went and did a bacterial smear first, and then you apply the stain. For a given length of time, you then pour off the dye, wash it off, dry it, and then examine the cells. A simple stain can use a mordant. We'll talk about that later. A mordant is not necessarily another stain. It simply intensifies the first stain, making it the cell more visible. So it increases the affinity between the stain and the specimen, or it could add to the first dye, increasing the affinity, coating the specimen to enhance it, thereby making it easier to stain. One mordant we can use is in the flagella stain, and it simply coats the flagella, and then you coat the flagella many times, and then when we look at it under the microscope, we can actually see the flagella. We'll talk about a mordant when we're talking about the Gram stain, because the Gram stain does have a mordant. All right, moving on to a differential stain. A differential stain reacts differently with different kinds of microbes, and therefore you can distinguish between two types of microbes. They usually will look different, such as they will stain two different colors. And to get the two different colors, we use two different dyes. Any question about any of that? So a differential stain usually uses two or more dyes. That is not required, though, that you have two dyes. Occasionally you'll have a differential stain that is only using one dye, and that dye just happens to stain cells differently, and so we can see differences within the cell even though we're only using one dye. And I'm sorry that's a bit confusing, but that's just the way life is, so you have to live with it. Sorry about that. The most common differential stains that we'll talk about are the Gram stain and the acid-fast stain. Those are the two most common differential stains. So the first differential stain, the Gram stain, was developed in 1884 by Hans Christian Gram. And you don't need to know the name, but you should know that Dr. Gram discovered the Gram stain, and so whenever you use it, you should capitalize the G in the Gram stain, because the stain is named after somebody. Okay? What I should do is penalize people when they don't capitalize it, but I don't do that. It's a very useful stain in bacteriology because it allows us to classify bacteria into two very large groups. The gram-positive cells and the gram-negative cells. Any question about any of that?
What do you capitalize?
Say again, I didn't hear that.
What do you capitalize?
The G in gram.
Okay, thank you.
Okay. Let me state a little bit about the Gram stain, and that is it was discovered by Hans Christian Gram. Initially, he made it so that it could differentiate between bacteria and the cells of the patient. So he was using it as a differential stain to distinguish human cells and bacteria cells. And the bacteria cells that he was doing were staining purple, and the human cells were staining pink. And so he was making it differential for that purpose. But when other bacteriologists or microbiologists started using his stain, they realized it was differential, separating all bacteria into two very large groups. The gram-positive cells stain dark or purple, and the gram-negative cells stain pink. And because of that, this stain is probably the most widely used stain in microbiology. Let me see if I want to talk any more about that. So Hans Christian Gram did intend it to be differential, but microbiologists made it differential for all bacteria. And Hans Christian Gram was looking at an infection of a patient by a gram-positive bacteria. All right, I think that's all I want to talk about with that. Let's move on to the next slide. The Gram staining procedure starts with a bacterial smear on a glass slide, meaning before you begin the Gram stain, you have to make a bacterial smear. And that bacterial smear has to be air dried and heat fixed. Because if you don't air dry it and heat fix the bacteria onto the slide, the bacteria will wash off with the washing step. And we have a lot of washes in the gram stain. I'm just highlighting, there's 3 of them in the 10-step process. And so the air drying and the heat fixing causes the bacteria to stick to the glass. And that keeps them from washing off. Any question about any of that? All right. So after you heat fix, air dry and heat fix the bacteria smear, you then flood the slide with Crystal Violet. And that is a stain. You flood it for 1 minute. And then after 1 minute, you dump off the Crystal Violet and wash with cold water to get rid of the Crystal Violet. And then you apply iodine for 1 minute. Iodine, in this case, is a mordant. And what iodine does is it complexes with the Crystal Violet and makes a bigger molecule. So the cells taking up the Crystal Violet will show a little more intensely because iodine Crystal Violet is a bigger molecule than just Crystal Violet. So iodine is a mordant. You then pour off the iodine and wash the cell with cold water. You do the wash gently, by the way. And then you remove most of the water. And now you flood the cell with acetone ethanol. And the acetone ethanol is a decolorizing agent. You decolorize for only about 5 seconds. The best thing to do is to hold the slide at an angle and then put on the decolorizing agent across the top of the slide and allow it to run down the slide. When you see the blue stop running, stop adding the decolorizing agent and then wash the slide immediately. And that way you're applying the decolorizing agent for the proper time. With the decolorizing agent, you want to remove the iodine Crystal Violet from the gram-negative cells, but not from the gram-positive cells. And if you leave the decolorizing agent on the cells for too long, it will pull out the Crystal Violet iodine from the gram-positive cells as well as from the gram-negative. And if you don't leave it on long enough, you will not pull out the Crystal Violet iodine complex from the gram-negative cells. So what you do is you observe the slide, apply the decolorizing agent, and once the blue stops running, you stop applying the decolorizing agent and then you wash it immediately. Now note how I'm saying wash immediately. I didn't put that anywhere else. You need to wash off the decolorizing agent because even if you dump off the decolorizing agent, it will be on the cells a little bit and it'll continue acting. And so you need to wash it off immediately to stop the decolorization. You then pour off the water and flood the slide with safranin for about 1 minute. Safranin is a counter stain because it's the second stain we're using and it has a different color than the Crystal Violet. So that's why it's called the counter stain. And the procedure I use is you use 1 minute for everything except for the decolorizing step, which is about 5 seconds. You then wash the safranin off, let the slide dry, and let me show you. Can you guys see this? I'll try and put this down so you can see this a little better. What you do is you get blotting paper, which is called bibulous paper, and it's thick paper, almost like cardboard, a little thinner than cardboard. It's almost like paper, cardboard. And then you fold the bibulous paper over the slide. Let me do this differently. So I use this for my slide and this for my bibulous paper. So I can fold it. And then you just pat it down. You do not move the slide or the bibulous paper like that. You don't move it over each other. You just blot. The reason why you're blotting is to remove the water. If you were to take the bibulous paper and move it along the slide, that will rub off the cells. And if you just blot it, that removes enough of the water so you can actually apply oil and then observe the cells under the microscope. Any question about any of that? If not, let's go into the gram stain in greater detail. When you do the gram stain, first you apply the crystal violet, then you apply the mordant. You'll note that the cells stain similar with the crystal violet, and you apply the mordant. The only difference is that the cells will be a little darker here, a little more purple. So this you could call a lighter blue purple, and this would be a darker purple purple. Then you use the decolorization step, and that does not remove the crystal violet iodine complex from the gram-negative cells, but it does remove the crystal violet iodine complex from the gram-negative cells. So before the application of the decolorizing agent, all of the cells were staining purple. After applying the decolorization step, only the gram-positive cells are purple, and then the gram-negative cells are clear. We then apply safranin, and that causes the clear cells to pick up the safranin and then stain pink, allowing us to differentiate between gram-positive cells, which are purple, and gram-negative cells, which are pink. Any question about any of that? All right, now let's talk a little bit about the differential properties of the gram stain. The crystal violet sticks to the peptidoglycan in the bacterial cell wall. It's the primary stain since it colors all cells the same. All cells stain purple. Oh, I got it there. All cells, the gram-negative and the gram-positive, stain purple. We then apply mordant, and it increases the affinity of the crystal violet for the specimen, and it complexes with the crystal violet to make a larger molecule. Once again, the cells stain purple. It's true they're a little darker purple and a little more purple purple, whereas before they were a little more blue purple. Let's see, is there anything else I want to talk about that? After the wash, you then apply the decolorizing agent. It washes the crystal violet iodine complex out of the gram-negative cells, but if it's done properly, It does not wash it out of the gram-positive cells. So this step is the differential step in the gram stain, allowing you to differentiate between the two types of cells. You need to wash immediately to remove the decolorizing agent off of the cells. And the decolorizing agent can be different substances. I think in the video that I'm going to show you in the lab, they're only using alcohol. Here we're using acetone and ethanol. You can also use acid and ethanol. Any time you use the decolorizing agent, you need to wash it off immediately to stop the decolorizing agent from acting. This is particularly true with acetone and ethanol and acid and ethanol because they react very quickly. And if you were to leave the decolorizing agent on the cells for any length of time beyond 5 seconds, like even 10 seconds, you'll start to wash the crystal violet iodine out of the gram-positive cells. But once again, this step is tricky because you do need to leave it on long enough to wash the crystal violet iodine out of the gram-negative cells. If you don't have it on long enough, you will not wash all of the crystal violet iodine out of the cells from a gram-negative cell. All right, any question about any of that? So this is the differential step where the gram-positive cells will retain the crystal violet iodine and they will be purple in color. The gram-negative cells lose the crystal violet iodine and they become colorless at this point in the gram stain. We then apply safranin. It's a pink dye. It does stain all the bacteria, meaning it does stain both the gram-positive and the gram-negative cells. It is a counter stain because it has a different color to the primary stain, the crystal violet, which is purple. And then we use safranin for 1 to 2 minutes. As long as you have a strong enough solution of safranin, you only need 1 minute. However, if your safranin solution is weak, you need to apply it for 2 minutes. And you just use it, and you know, is your safranin good or is it weak? You then pour that off, wash the slide with distilled water. Let it dry and examine the cells, and the cells that are gram-positive stain purple from the crystal violet and iodine, and then the gram-negative cells stain pink from the safranin. And I've already talked to you, I had to blot with bibulous paper the slide so you can observe it microscopically. All right, let's come back here. I stated that safranin gets into the gram-positive cells. So why aren't these cells staining pink? Somebody can guess this. Why are the gram-positive cells not stained pink?
Is it a mixture of the safranin stain and the iodine causing it to turn purple?
It is a mixture, and it is true that if we were to look at the cells with an electron microscope, so let's make this, for example, a cell, this box here, we could see with the electron microscope a region of the cell staining purple, and there'd be more than one region. I'm just giving this as an example, okay? So my point is you'd see purple spots in the cell, and you could see pink spots in the cell with the electron microscope. So the safranin is in the cell. However, when we look at the slide with our human eye, the purple is more intense, and that's the only color that the human eye sees. It does not see the pink, and the purple just, what do you call that? Overshadows the pink. And so when we look at it, we can't see the pink in the cells. Whereas the gram-negative cells, they only have the pink specks in the cell when we look at the cell with the electron microscope. And so our human eye, when we look at the cell, it will appear pink. Okay, so the safranin is in the gram-positive cells. It's just that our human eye cannot see it. And to see it, you can't use a light microscope. You have to use an electron microscope and then magnify many times. Any question about any of that? So what is the difference for the differential staining? What's the basis? And I briefly mentioned it earlier. It has to do with the differences in the cell wall. The gram-positive bacteria have a very thick peptidoglycan layer. We'll talk more about peptidoglycan, its disaccharides and amino acids. And this very thick peptidoglycan layer traps the crystal violet iodine, and it does not allow it to wash out with the decolorizing agent being applied for 5 seconds or about that time. Okay? In the gram-negative bacteria, they do have a peptidoglycan layer, but it's thin, much thinner than the thick peptidoglycan in the gram-positive. And they do have a lipopolysaccharide layer as the part of the cell wall. And this thin peptidoglycan layer is not thick enough to trap the trap the crystal violet iodine. And so it washes out of this thinner peptidoglycan layer. So when we're looking at the gram-positive cell, that's the cell here, the purple being the cell wall, the yellow being the cell membrane. And when we blow that up here, yellow being the cell membrane, purple being the cell wall, actually all of the cell wall of the gram-positive cell is peptidoglycan. And it's not 3 layers, this is just an artist's rendition. It's layer upon layer upon layer, hundreds or thousands of layers of peptidoglycan, many, many layers. Okay? And there's the cell membrane. It's below the cell wall. This is what the gram-positive cell looks like. A very thick cell wall that's mostly or entirely peptidoglycan, and then the cell membrane. And then the crystal violet iodine gets stuck in this very thick peptidoglycan. The gram-negative cells shown here have a different cell wall. And so when we look at the cell, it does have the membrane, that's the same. And it does have the thin peptidoglycan layer, which is that thin purple layer right there. And then it has a lipid membrane on top, which we'll talk about a little bit later, but that's the outer membrane of the cell wall. And it's similar to the cell membrane. So it is a membrane. There is a thin peptidoglycan layer, and in this artist's rendition, it made the peptidoglycan layer only one layer thick. That's not correct. The peptidoglycan layer is many layers, let's say around 20 to maybe 100 different layers of peptidoglycan, but it's much less than the layers of the gram-positive cell. Okay, so it's not one layer of peptidoglycan. It is layer upon layer, but it's many less layers of peptidoglycan than in the gram-positive cell. And like I said, the best example will be to think of it as laying 20 layers of peptidoglycan. Any question about any of that? Oops, where do I have that? So the crystal violet iodine complex forms in all cells, and the crystal violet iodine complex is larger than the crystal violet or the iodine alone. And the crystal violet iodine complex is washed out of the thin peptidoglycan layer of the gram-negative cells, and the lipopolysaccharide layer of the gram-negative cells is destroyed by the ethanol of the decolorizing step, meaning ethanol destroys the lipid gram-negative cell outer cell wall as well as the cell membrane. And that means there's nothing to keep the crystal violet iodine complex in the cells, and so it washes off. The cells are therefore colorless. The gram-positive cells, the crystal violet iodine cannot wash out of the thick peptidoglycan layer, and therefore the cells retain their purple color. And then when we look at cells under the microscope, the cells will stain different. The gram-positive cells right here will stain purple. There's a purple cocci, another purple cocci. And then the gram-negative cells will stain pink. There's a pink spirilla. There's a pink rod. There's a pink vibrio or bent rod. That looks like a pink cocci. So these are all gram-negative cells, and the crystal violet iodine washes out of the gram-negative cell, at least if the stain is done correctly. Any question about any of that? All right. Now the gram stain is a little more hairy than when we talked about it. And so let's talk a little bit about the complications of the gram stain. Like this is a complication of the gram stain. What's going on? Where do we do the gram stain? Because these cells are definitely lighter in color than these cells right here next to the crystal. And actually this is not cells. What this is a crystal of crystal violet, and the crystal violet solution, the crystal violet will precipitate out of solution and form a crystal, and in the bottom of the bottle there will frequently be crystals of crystal violet in the crystal violet solution. Let's move on. So where do you do the gram stain? Well, you don't do the gram stain in the bad region, which would be here and here and here. You do the gram stain in the good regions of the slide, where it's easy to distinguish individual cells, and the color is good. Meaning these cells all have about the same color. These cells Yeah, these are a little bit lighter than those, but generally they're similar. It's different than right here, where these cells are much darker than those cells. So, some complications of the Gram stain. To get consistent results, when you do the Gram stain, you must do the Gram stain on young growing bacteria. If the bacteria are over 2 days old, do not do the Gram stain on them, because the Gram stain may not stain correctly if you have old cells. It is true that some bacteria cells stain poorly or not at all with the Gram stain, so it may not be universally applicable, and usually it's the Mycobacterium which stain poorly, and we'll talk a little bit more about those cells in a little bit. They resist taking up the water-soluble dyes, the Mycobacteria, and so they don't stain too well with the Gram stain. The results are not always clear-cut as the theory suggests, and this will become evident during your unknown project, where it looks like the cells are one color, but in reality the Gram stain was not done appropriately. Perhaps let's go back to that slide. So if you have old cells, the old cells may not take up and retain the crystal violet correctly. Old cells tend to die, and so they'll have holes in their cell wall, and then the crystal violet and the iodine can come out of the cells. Another thing is if you have too thick of a layer of bacteria, it'll be difficult for the crystal violet iodine to get through the cells to stain the bottom layer of cells. Or another problem of too thick of a layer of cells is with the Gram-negative cells, you will have trouble washing out the crystal violet iodine from layer upon layer of cells. So that's why you want a good thin bacterial smear where ideally you have the bacteria in only one layer of cells. Anything else I need to say about that? If the bacteria are very thick, you'll also have trouble washing out the crystal violet from the lower layers. You shouldn't have any trouble from the outer layers, but the lower layers, you'll have trouble. So that will be a problem with the Gram-negative cell. And the problem with the Gram-positive cells, if you have layer upon layer of cells, you have trouble getting the crystal violet iodine inside. The first layer will stain nicely, but lower down you have trouble getting the cells to stain purple. So that's why you want a thin bacterial smear when you're doing the Gram stain, and always use cells that are 2 days old or less, meaning newly growing cells. And then when you're looking at the slide, do the Gram stain on a region of the good part of the slide, not a region of the bad part of the slide. And I have seen this before on a slide where there's a crystal violet crystal, and that crystal tends to make the cells in the region near the crystal stain purple, even if the cells are Gram-negative, and further away from the crystal, they're staining pink. Any question about any of that? All right. So those are some complications of the Gram stain. The Gram stain is tricky. And generally, it takes practice. You have to do it a couple of times before you become good at doing the gram stain. All right, the structure of the gram-positive and gram-negative cells not only affects the staining, determining on whether they're stained gram-positively or gram-negatively, it also relates to how the cells respond to many antibiotics. Gram-positive cells tend to be killed easily by penicillin G and a bunch of other antibiotics like cephalosporin, penicillin V, many detergents. They're easy at killing gram-positive cell. The gram-negative bacteria, however, are more resistant to these antimicrobials. Penicillin G cannot kill a gram-negative bacteria. Many detergents do not kill gram-negative bacteria. The trouble is that the penicillin G and the detergents cannot penetrate the lipid polysaccharide layer of the gram-negative cells. Let me come back here. Right here. Penicillin G cannot get through this outer membrane of the cell wall. And so penicillin G can't get into the cells. It will not harm the gram-negative cell, but it can get in the gram-positive cells, and it will harm the gram-positive cells. Penicillin G is the only antibiotic I will discuss this way, and that doesn't mean that gram-negative cells are harder to treat than gram-positive cells. We just have to use another antibiotic besides penicillin G. We can even use ampicillin to kill gram-negative cells. And ampicillin is one of the early members of the penicillin family. Okay, I'm not sure what number it is. It might have been the third member of the penicillin family discovered, but if it wasn't the third, it was within the top 10. Okay, and there's over 40 members in the penicillin family. And ampicillin, it was a very early member of the penicillin family to be discovered. You can also use other antibiotics that can get right through this outer membrane of the cell wall. Any lipid-soluble antibiotic can kill gram-negative cells, and I'm drawing a blank here for what I'm trying to think. Streptomycin is an antibiotic which goes right through the bilayer of an outer membrane of a cell wall, gram-negative cell wall. So streptomycin can kill both gram-negative cells and gram-positive cells. It's the same as ampicillin, whereas gram-positive cells are killed by penicillin G, which penicillin G does not kill a gram-negative cell. Any questions about any of that? All right, let's talk briefly about special stains. You're not going to be examined on this. However, you need to know about special stains for your unknown project. Like the capsule stain is a special stain, and this stain is used to stain capsules. Some of you will need to do the capsule stain for your unknown project to determine whether your cells have a capsule or not. The capsule stain stains the background of the cell and the cell, but not the capsule. Okay? So this right here the clear region around the cell, which is kind of black or very, very dark purple, is the cell. And then the capsule is clear, or that kind of looks a light blue. And then the purple is the capsule stain staining the background of the slide. This slide says this is a negative stain. This is not a negative stain because I just know it's not a negative stain. This is a capsule stain, but not a negative stain. A negative stain is putting on a dye which stains the background, and it may stain the cell slightly, like this iodine. India ink slide is a capsule stain that is a negative stain. And see how it's staining the background very, very darkly? And it is lightly staining the cells, but then the capsule is not being stained. And so this clear region is the capsule of the cell, and there's the stain there, which is like a grayish color. And then the background is so dark it's almost black. This is India ink, and it just stains this way. You're only using one dye here, but yet you can see the cells, the capsule, and then the background. This stain is probably using two dyes, one to stain the cells, and then another dye to stain the background of the cell. And then the capsule is not being stained. All right, any question about negative cells? If not, that's it for chapter 3. Are there any questions about chapter 3? If not, let's move on to chapter 4.
I had one question really quick.
Ah,
And you may have answered this at the beginning of the class. I was like 5 minutes late, so I don't know if you did. But the quiz for Wednesday, like for both quizzes, they don't cover chapter 4, right? Just up to
that's
chapter
correct.
3.
Let me turn you up. I'm barely hearing you. The syllabus tells you what the quiz is covering. The quiz
Okay,
is
perfect.
covering chapters 1 and 10, and only the start of chapter 2. So the material we covered last Thursday will not be on the quiz. Only the start of chapter 2, not the end of chapter 2. And the quiz also has lab 0, lab 1, and lab 2. So none of chapter 3, and the end of chapter 2 is on quiz 1. Okay?
Okay, got it. Thank you.
All right. So chapter 4, functional anatomy of prokaryotic and eukaryotic cells. We will definitely talk about prokaryotic cells, but if I do not finish talking about eukaryotic cells, I'm just going to assign that to you guys because your preliminary class, which was Biology 160, was supposed to talk to you about the anatomy of eukaryotic cells. So this should be only a review, and if we do not finish it, I'm just going to assign it. Okay? Any question about any of that? All right. The major goals on the rough outline are discussed here. Know the terms. What's a coccus, a bacillus, what's a sporilla, a strepto, like a streptococcus, a staphylo, which is always staphylococcus, a diplo, which is like diplococcus. What's a tetrad? What's a sarcenia? What's a vibrio? what's a spirochete, what's the glycocalyx, and you should know there's 3 types of glycocalyxes, and we'll talk about that. What's a flagella, what's an axial filament, what's a fimbriae, what are pili, what is peptidoglycan. We've kind of talked about that, but we'll go into it again. What's endotoxin, what's selective permeability, what is diffusion, facilitated diffusion, what is osmosis, what is active transport, what is tonicity, what's a plasmid, what are inclusions, what are endospores, and then lastly, we'll talk about the endosymbiotic theory in this lesson. And if I don't do any of the eukaryotic cells, I will talk about the endosymbiotic theory. Okay. All right. So in this lesson, we'll also talk about how gram-positive and gram-negative cell walls differ, and you need to be able to describe the importance of taxis in bacteria. Any question about what we're doing? All right, so you should know that prokaryotes literally is Greek for pre-nucleus. Specifically, this is pre-kernel, and they're using kernel for the nucleus. Eukaryotes is Greek for true nucleus. So U means true, and karyotes means kernel, and we're using the kernel for the nucleus. So the eukaryotes have a true nucleus. The prokaryotes do not have a nucleus. The chromosomes also differ, at least in the bacteria, and that is that bacteria have 1 circular chromosome. It's not in the nucleus, whereas the eukaryotes generally have many linear chromosomes, and they are within a nucleus. I think we talked about the circular chromosome before, and that is, it's a circle, so that wherever the chromosomes start, that's where the chromosome ends. In a linear chromosome, like the X chromosome, there's a beginning place and an ending place. And the ending place is not the same place as the beginning place. I think we briefly mentioned that the DNA of bacteria are not associated with histones. We're not going to talk about the archaebacteria, but with bacteria, their DNA does not have histones. And then the DNA of eukaryotes is associated with histones. And the histone is the main protein in the eukaryotic chromosome. There are other proteins, but the histone is the main protein. Prokaryotes, besides having no nucleus, they have no organelles. And when I mean organelle, I mean membrane-bound organelle. The nucleus actually is a membrane-bound organelle, but the prokaryotes have no other organelles like mitochondria, chloroplast, Golgi complex. So they're not seen in prokaryotic cells. They are seen in eukaryotes, which do have membrane-bound organelles. When we look at the bacteria, they do have peptidoglycan and their cell walls. And if the bacteria has a cell wall, peptidoglycan is the main molecule in the bacterial cell wall. In gram-positive cells, I think there's only about three molecules in the cell wall. And of course, peptidoglycan is the most common. Even in the gram-negative cells, peptidoglycan is the most common molecule in the cell wall, if the cell has a cell wall. All right, the eukaryotes have no peptidoglycan in the cell walls, And of course, animal cells do not have a cell wall. Prokaryotes mainly divide by binary fission, where the cell here, when it's ready to divide, will literally split into two, and then each half will become a new daughter cell. Eukaryotes generally do not divide that way, and when eukaryotes divide, they divide by mitosis, where the cell goes through mitosis before dividing. All right, so this is a summary. We're going to talk about the prokaryote cell shape and arrangement, then move on to the cell structures, and then movement of materials across the cell membrane. If we have time, we'll talk about the cell structures of eukaryotes, and then at the end, regardless of whether we have time or not, I will discuss the endosymbiotic theory. And that will be not today, but sometime in the future, maybe our next lesson. All right, prokaryotic cell size. They are smaller than eukaryotes, and most cells are between 1 and 3 micrometers in size. You can get cells, prokaryotes, up to 10 micrometers, and that'll be the length, meaning they're not usually that long in the diameter. Prokaryotic cells basically take 3 main shapes. A coccus cell, which is round, bacillus, which is a rod. Let me blow that up. Right there would be a better one. A rod right there. And then the third basic shape is a spiral, sort of like an S. And these are the 3 scientific names: coccus, bacillus, spiral, or spirilla. Plural for coccus is cocci, and you can pronounce that differently. I think I've got that in another slide. See if I can remember. There's 4 ways you can say cocci. Cocci, 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 and I've forgotten the third way. Cocky. So, cocci, cocky, cocci, and I forgot the 4th one. I gave you all 4 of them though. I lost my count, so I wasn't sure what the 4th one was. The bacillus are also called rod, so both of those are scientific terms, and you can call a bacillus a rod and a rod a bacillus. The plural for bacillus is bacilli. Rod is singular, rods plural. Let me see if I can remember. Spirals and spiral is singular and spirals is plural. And spirilla and spirillum. So spirillum is singular, spirilla is plural. Hopefully I got that right. The first slide I'm going to talk about different types of spirals, and the first is a vibrio, which is a half a spiral. You could also call this a bent rod, and I typically call a vibrio a bent rod, or you could call it a vibrio, okay? A curved rod. I'm trying to remember the bacteria. Vibrio, I'm drawing a blank now on the name of a Vibrio, but it's fine because we're not going to really talk about that disease. But there is one disease which is caused by a Vibrio, and I'm forgetting the name of it now. It'll come to me, in which case I'll state it. Aspergillum. You have two types. Aspergillum has just a few bends, so more or less an S-type shape, and it's a rigid cell. A spirochete has multiple bends, and it's more flexible, so the cell can literally bend its shape. Both of these cells can move with a corkscrew type movement, and I'll show you that in a little bit when we're talking about the movement of the spirochetes. I think I've got a picture of it, or a movie of it. Any question about the different types of spirals? All right, as I stated, bacteria, including most prokaryotes, have three main shapes: the coccus, the bacillus, and then the spirals. But there are a few prokaryotes that have unusual shapes, like the Stella bacteria is actually star-shaped, and then the Haloarcula is square or rectangular shape. Very few bacteria are unusual in shape. Most bacteria species are monomorphic, meaning they take on a single shape, like Horcula, shown here. All of them are rectangular in shape. The Stella, oops, missed too far. All of them are star-shaped. So that's monomorphic, where the bacteria take on one shape. There are a few species, though, that are pleomorphic, where the cells will take on many shapes. And here we have a bacteria that is pleomorphic. Let me try and blow this up a little bit. Too far. There we go. Well, you can see part of it here. Different shapes. This one is almost cockeyed. That one's more cockeyed. That one's sort of dumbbell shaped. Same with that one. And that one, I don't know, a bent rod. No idea what that shape is. Almost like a figure eight. Pleomorphic, where the cells, this is one species, and the cells are pleomorphic, taking on many different shapes. Any question about any of that? All right. So let's talk about the cellular growth arrangement. This is the way that cells stick together when they divide. This is the growth arrangement, or the cellular growth arrangement. The cells that stick together take on a shape, and the different shape we can call singlets. If all the cells are single cells, it's singlets. There can be diplococci when two cocci stick together, shown here, right there. There can be streptococci, and that's when the cells divide all in the same plane, and so this cell divided here, that cell divided there, that cell divided there. There are tetrads, and that's when 4 cells stick together. Sarcenia, that's when 8 cells stick together. And the sarcenia, the cells divide on all 3 planes, making a sarcenia. If they only divided on 2 planes, we'd have a tetrad. If they only divide on 1 plane, you'd have a diplococci or a streptococci. And then if the cells divide on all planes, we have staphylococci. Staphylococci literally means a cluster looking like grapes. So a cluster of round cells, cocci cells, in a cluster like a grape. All right, these are all the different shapes you can see with cocci. And do note that a singlet isn't on here, but it is a type of cell. So we got singlets, diplococci, streptococci, tetrad, Sarcenia, and staphylococci. With the bacilli, their cells take on fewer cellular growth arrangements than the cocci. First, you can get a single bacillus, so all the bacteria growing as single bacilli, and those we call singlets. Most of these are singlets. You can have diplobacilli, where 2 bacilli stick together. And I don't see that, although there could be. Where was it? Right there, that might be 2 stuck together like a diplobacilli. That could be 1, it's hard to say, especially with my eyesight. Streptobacilli is where they stick together on the long end of the cells. And there we have streptobacilli, there's a bacilli, there's a bacilli, there's a bacilli. If you have the streptobacilli with the cells stuck together on the long end, we don't call it streptobacilli, we call it palissae. A fence of rods. Let me see if I can draw that. Okay, I got it. So if the bacilli, don't have it. Why don't I have it? Oh, there we go. Now I'm just going to draw it like this. This we call a palissae, where the bacilli are stuck together on the long end. Why it's called the palisade is we have fences which have boards of wood, and the fence looks like this. And if you read Mark Twain, Tom Sawyer was painting a palisade fence. Okay? Palisade fences aren't as common nowadays, but especially in the 1800s, the early 1900s, they were very common. You can still see them occasionally today. All right, any question about any of that? So those are the arrangements of bacilli. Singlets, diplobacilli, streptobacilli, and palisades. You do not have the other arrangements that you have in cocci. Any question about that? All right. Let me talk briefly about here. This is a shape, which is, oops, not too far. Coccobacillus is a rod that you shorten and has rounded ends. And you shorten the rod until it's almost a cockeye. And these right here are coccobacilli. When we look at this, it's hard to determine, is this a coccus? Coccus, or is it a real short rod? And this we give the term coccobacillus. Even that one there is a coccobacillus. And that's a different shape than the three main shapes we discussed. But the coccobacillus is one of the three main shapes. It's just a combination of a cockeye and bacilli. You should note the bacillus has two meanings. If it has a capital B, that means it's a family of bacteria. And if it is a lowercase b, it means that it is a shape of bacteria. Okay, so the family Bacillus, we always capitalize. And then the shape of bacteria, we always use a lowercase b. With Streptococcus, don't confuse it with Streptococci. There is a family called Streptococcus, and that is a family of bacteria. Streptococci is a chain of cells, and the good news is the family of Streptococcus does have the shape Streptococci. Okay, not the shape, the cellular growth arrangement. All right, now there's another complication I need to talk about with the shape, the cellular growth arrangement, and that is if you have a cellular growth arrangement other than a singlet, you'll always have more than one cellular growth arrangement. And that's because the cells can break off. And so if you have streptobacilli, you will see, if it breaks there, you'll see in the slide streptobacilli, you'll see diplobacilli, and you'll see singlet bacillus or singlet bacilli. And so when you're reading the cellular growth arrangement, you always take the highest order. Is that clear? That when you look for the cellular growth arrangement, you'll always see more than one cellular growth arrangement, unless there is only singlets. Because if there's only singlets, that's all you'll see, singlets. But all the others, you'll see more than one cellular growth arrangement. Even if it's diplobacilli, occasionally that'll break, and you'll see just diplobacilli and singlet bacillus. That doesn't mean you'll see all cellular growth arrangements. For example, if you have streptococci, you can see diplococci, and you can see singlet cocci, but generally that's all you might see. You might not see tetrads, you might not see Succinia, you might not see staphylococci. Okay, so you might not see all of them, but you will see more than one, unless you're only seeing singlets. And I guess the other thing I should mention is with the spirals and the spirochetes, they only take on one cellular growth arrangement, and that is singlets. these cells do not stick together. All other cells do stick together, and usually we can see more than one cellular growth arrangement. Any question about any of that? All right, if not, let's briefly talking about the structure of a typical prokaryotic cell, and then go into the different cellular structures that we see in prokaryotes. So here is an actual electron microscopic image, and there's the artery rendition. You can see the nucleoid region stands different than the cytoplasm. However, in a prokaryote, the nucleoid region is wherever the DNA is floating around in the cytoplasm. And that DNA can float anywhere in the cytoplasm. There is no distinction between the nucleoid region and the cytoplasm because there is no cell membrane separating the nucleoid region from the cytoplasm. It's wherever the DNA is, that's where the nucleoid region is. And that nucleoid region just stains a little differently. Why we call it the nucleoid region. All cells tend to have the red structures. So all cells have the plasma membrane, also called the cell membrane. All cells have the cytoplasm, and all cells have DNA. I'm going to word it DNA, and bacteria have a nucleoid region, but eukaryotes have a nucleus. And we're just going to say all cells have three characteristics: DNA, the plasma membrane, and the cytoplasm. All cells also have ribosomes, but ribosomes are found in the cytoplasm. And they're large enough, the ribosomes are large enough structure so that they are not in solution of the cytoplasm. And so you can actually see the ribosomes here. Let me see if I can see it here. There are the granules here. Our ribosomes are the regions where proteins are synthesized in the cytoplasm. Cytoplasm is just the liquid portion of the cell outside of the nucleus, and prokaryotic cells do not have a nucleus. So cytoplasm is really everything of the prokaryotic cell. All right, I've talked about that. Let's talk a little bit more about the cell wall and the cell membrane. So the innermost region of the cell wall is actually not the cell wall, it's the cell membrane or the plasma membrane. Then it's a lipid bilayer. We'll talk a little bit more about that. Outside of that, we have the cell wall. Outside of the cell wall, which is the last portion of the cell. The outermost portion of the cell is the cell wall, but outside the cell wall, we may have a capsule. And it's something secreted by the cell, and it's external to the cell, external to the cell wall. And we'll talk a little bit about the capsule in a little bit. The fimbriae is something that's attached to the cell wall. We'll talk a little bit about that. The same with the pilus. Inclusions I think I have a slide talking about that, is something in the cytoplasm that is accumulated to large enough portions that it actually precipitates out of solution. All right, any question about any of that? I've talked about everything except for plasmids and flagella. Plasmid is a short piece of DNA that is outside of the chromosome, and that we call a plasmid. All right, any question about this slide? If there's no further questions, I think I'm going to stop here and I will see you. Is it supposed to be 6:50? Or is it 7 o'clock?
It's 6:57 is when our lab starts.
Yeah, 7. All right. So no, I'm going to stop here and then I'll start the lab at 7. All right, any questions? If not, I'll see you in the lab at 7.
Thank
Thank
you.
you.
All right.